Alright, let's see what you guys have retained. Got a piece of paper, write your name in the corner. What the heck? First question. What is DNA? I'm just giving this away.
Sorry Valentine, what is this out of?
Maybe 0.25. Next question. A baby hamster's kidney cell has a surface area of 3,400 micrometers squared. Assume that the endocyte clathrin-coated pits account for 2% of this area, and that the surface area of a single coated pit is the same of that of 100, I don't know, I guess I forget this measurement, but nm, diameter clathrin-coated vesicle that will be formed from it. If the cell surface has 100,000 transferrin receptors, around 70% are localized within coated pits. Work out the following. If a transferrin receptor has a cross-sectional area of 50 angstroms times 100 angstroms, how many receptors could be accommodated in 100 nm clathrin-coated vesicle, assuming that only 50% of the surface area can be occupied by proteins? Please express the answer in binary, or I will give this one away. Latin. Get out! See ya. We haven't learned anything of that magnitude. You should be reading ahead! Oh yeah, you got exactly 180 seconds left. Oh God, I wish I could menstruate.
Pardon me?
Do your work! If I could menstruate, I would be more in tune to the tides, and I would not have to deal with idiotic calendars that I could just count down from my last cycle. And that's time. Pass your tests over. What did you get for the first question? Yes, I want to say Prickly Pete.
Kai.
I don't care. I'm reducing you all to numbers, okay? 1, 2, 3, 4, 5, 6. Okay? Remember your numbers. Number 2.
Called deoxyribonucleic acid and is comprised of nucleotides.
Is that all?
Yeah, that's all I wrote.
Put weakness, exclamation mark, then idiot within question mark. Actually, no. Who wrote that? Put up your hand. Raise your hand. Number 2. Get the hell out! Don't help him. He's a failure. Leave your things. They're mine now. That's right. Get out. Good riddance. Waste of my time. Waste of flesh. What did you get, number 3?
Irradiate the hypersinium miscamercephalus. This will enable BKH to be more clearly visualized.
That is only so-so. But since I'm such a nice guy, I will give it out of 1 out of 6. Back to DNA translation. DNA helix unwinds. Transcription starts at the promoter. It continues until an entire gene has been converted to mRNA. New mRNA strands separate from DNA and goes to the cytoplasm, where it is used as a blueprint for the construction of a protein molecule. RNA is generally modified before passing out of the nucleus. These modifications are most characteristic of messenger mRNA, which provide the exact code for protein synthesis. mRNA can be anywhere from 250 to 10,000 nucleotides. Oh yeah, get out your homework I assigned.
What homework?
The 6 pages I assigned out of that green biozone that was supposed to be due 5 weeks ago. If you're not done it now, you're just useless.
Mr. Valentine, is it okay if I do and hand mine in next class? I have a spare.
Yeah, it's okay. If you can get out! Get out! Useless! tRNA and little r RNA constitute the cell's mechanism for protein synthesis. Ribosomes are assembled within the nucleus as two little r RNA protein subunits are just able to pass through a nuclear pore. I expect you all to draw this diagram with just with a lot of detail. What the heck? All of it? Yes, in detail. If you miss a single nucleotide, I will give you a huge wedgie.
I don't really understand translation. Could you just quickly go over it?
Shut up! I knew I was going to lose the weak ones, but we're moving on.
Action.
If I read this?
You don't read it, you just click past it.
Okay. It is the act of which the sequence of bases in mRNA molecule is used as a code for the specific sequence of amino acids in the protein molecule. RNA bases, uracil, which is pretty much, I guess, represented by a U, and adenine, which is represented by an A, and cytosine, which is represented by a C, and guanine, which is represented, you guessed it, by a G. mRNA has moved from the nucleus to the cytoplasm, and the ribosome has bound to it at a site consisting of 6 bases in a row. tRNA molecule binds at the first 3 bases at the second site. Another tRNA brings a different amino acid. This process repeats until protein is formed. Amino acids attach to the 3 end of the specific tRNAs. Both ribosomes, subordinates, latch onto the strand of mRNA. Within the ribosome, tRNA matches with mRNA. Really, guys, come on. Here are the steps. It goes with you.